Good morning, Priya. Good morning. Good morning. Good morning, all. Okay, good morning, everyone. Good morning. Okay, so friends, in the last class, okay, good morning, all. Good morning. Good morning. So, in the last class, we completed the translation in case of a prokaryotes also and eukaryotes also, and I have given some of the antibiotics that are blocking the process of translation. Is it? Yes, of course. Now, friends, we have to read the another very important topic. That is what do we can call it as post-translational modifications. Good morning, everyone. So, friends, everyone listen to me very carefully. We all know that. Just quickly recall whatever the mRNA that we have synthesized in the eukaryotic organism, that mRNA is not biologically active and that mRNA is not functionally active. Good morning, all. So, to make that particular mRNA biologically active, to make that particular mRNA functionally active, we have done certain processes like 5' capping, 3' polyadenylation, as well as the splicing. And if you all quickly recall that all those processes are occurred after the transcription. Good morning, all. So that's why we can call it them as post-transcriptional modification. Same way, friends, everyone listen to me carefully. Whatever the process of whatever the protein synthesized in the translation, friends, that protein is not functionally active, that protein is not biologically active. So we have to make that particular protein to become functionally active. Good morning, all. To become functionally active, to become biologically active, so for that we have to do certain processes. That processes are happening after the translation. That is the reason, friends, we will be calling them as post-translational modifications. Please kindly don't forget this point at all. So, friends, whether it is the prokaryotic organism doesn't matter, whether it is the eukaryotic organism doesn't matter. Where does this particular process of post-translational modifications occur? Of course, friends, the process of post-translational modifications occur exclusively in the cytosol only. Don't forget it. I will be explaining you briefly. Just listen to me carefully. Good morning, all. Good morning. Okay. So, good morning. So, friends, listen. Whenever a particular protein gets synthesized, everyone focus. We can call it them as the nascent polypeptide. So, friends, here the word nascent polypeptide means the polypeptide which is not biologically active, the polypeptide which is not functionally active. So all of you listen to me. The nascent polypeptides must be folded to their native conformation. So here what is the word native conformation, friends? Every one of you remember very clearly. Good morning, all. Good morning. Native conformation means, friends, biologically active conformation. We will be calling it as the native conformation. In a simple way, friends, we have to convert the nascent polypeptide into biologically active polypeptide. So how we are going to do? What are the various type of bonds are involved? Good morning, all. Good morning. What are the various kinds of bonds that are involved in this particular post-translational modifications? As we discussed, hydrogen bonds, van der Waal, ionic and hydrophobic, then followed by the disulfide. So everyone remember, friends, these five are the major forces or the major bonds that are involved in, good morning, that are involved in this particular process of post-translational modifications. So friends, let me tell you one simple example. You know, right, whenever we synthesized insulin, good morning all, good morning, whenever we synthesized the insulin, what do you call it insulin, friends? We call insulin every time as the pro insulin. See, whenever it's synthesized immediately, we never called it as the mature insulin, we called pro insulin. What do you mean by the word here pro insulin? Inactive form of the insulin. So friends, continue everyone. We are doing the post translational modifications. Please continue. Now this particular proinsulin, which is biologically inactive form of the insulin, we have to convert them into the mature insulin. So friends, how can we convert this particular proinsulin into the mature insulin, friends? Of course, by the process of post-translational modifications. Okay, so I want you to majorly remember, friends, the type of forces that are involved. What forces are there? Hydrogen bond is there, van der Waal is there, ionic, hydrophobic and disulfide. Okay, now listen to me carefully. What are the examples of post-translational modifications? So friends, everyone listen to me very carefully. Whenever we are reading the post-transcriptional modifications, what we read, friends? We read 5' capping, we read 3' polyadenylation, and finally, friends, we read the splicing. Is it? So these things we will be calling it as post-transcriptional modifications. So friends, there are many kinds of a post-translational modifications are there, friends. But among all, the most important type of post-translational modifications are proteolytic cleavage, covalent modifications, protein splicing. You know, right? In the when mRNA always try to correlate, it become very easy. When we are reading the mRNA splicing, we read that there is a formation of introns and exons. Same way, friends, there is no much difference. Here we call them as inteins and exteins. This is not greatly important, friends. If you can remember, otherwise leave it. But what I want every student has to remember is first proteolytic cleavage and covalent modifications. Friends, they are very important for the part B, even part C also. Most of the time we will get the questions from the part B. Okay, let's start the first one. Proteolytic cleavage. So friends, what you have to understand by the word proteolytic cleavage? Everyone listen to me. Proteolytic cleavage is the most common type of the post-translational modifications. I will tell you, friends, many examples of the proteolytic cleavage, many examples, and I want you to focus very clearly. I hope, friends, every one of you quickly recall, whenever we are studying the prokaryotic translation, every student remember, whatever the methionine, the first amino acid, that particular methionine added by a group, what is that particular group? That is only we can call the pharmyl group. So that's why I hope every student remember, we call it as the pharmyl methionine. Now you only tell me, friends, is there any significance? Understand everyone. Is there any significance or is there any biological activity of this particular pharmyl group? No, not at all. There is no biological activity of the pharmyl group. So friends, once the polypeptide gets synthesized, everyone listen, once the polypeptide synthesized completely, we have to remove this particular pharmyl group. How? By the process of proteolytic cleavage. In one line, I can define to you, proteolytic cleavage is a process, friends, where inactive precursors are getting activated under proper conditions by a process called as limited proteolysis. Let me tell you an example. How can we convert inactive precursors into active precursors? Every one of you pay attention, utmost importantly. See friends, whenever insulin got synthesized, how does the insulin can synthesize it? Friends, everyone pay attention. Insulin can be synthesized as the pre-pro insulin. What do you mean by the word pre-pro insulin? It is not at all biologically active. I want every one of you remember the pre-pro insulin, it contains chain A and it contains chain C and it contains chain B. So this is the chain A and this is the chain C and this is the chain B, and I want every one of you remember friends, this also contain, what does it also contain? A signal sequence. Please kindly don't forget that. So what is present friends? Signal sequence. We read in the cell biology chapter many times. Anyway, what is this particular signal sequence will do? This particular signal sequence always help. What it is helping? This particular signal sequence will help this particular polypeptide to reach the destined area. But you only try to understand friends. Do you think the signal sequence having the biological activity? Not at all. So friends, remember every one of you, by the proteolytic process, what we will do friends? What cleavage? By the proteolytic cleavage, we remove this particular signal sequence. Simple point friends. Upon removing the signal sequence, what is happening friends? The pre-insulin can be converted into what insulin friends? Pro-insulin. Now you only tell me, I have given you a hint. The pro-insulin is also not a mature insulin. The pro-insulin also not a biologically active insulin. So what we have to do? We have to cut this particular C chain also, and we will add chain A with the chain B with the help of disulfide linkages. So when we do this particular process, the pro-insulin can be converted into what friends? We can call it as the mature insulin. So friends, please kindly don't forget. See, look into this. This is in the diagram. With red color I am drawing something. That is what? Signal sequence. So when there is a presence of the signal sequence, we will be calling it as the pre-pro insulin. Upon removing the signal sequence, what are present friends? Chain A, chain B, chain C. And please friends, every one of you listen. Chain A consists of 20 amino acids. Chain B consists of 31 amino acid. And friends, chain C consists of 33 amino acids. So total how much friends? Total 84 amino acid. What you people has to remember? The pre-pro insulin consists of total 84 amino acids. So we will cut the signal sequence. Pre-pro insulin only converted into mature insulin. How? By the cleavage of signal sequence and by the cleavage of the C chain and joining of A chain and B chain with each other. And what happens to this particular C chain or the C polypeptide? The C polypeptide will get completely degraded. Very simple point it is. So this is what I am explaining you here. I am explaining you the first type of a post-translational modification, that is the proteolytic cleavage, and I have explained you the first example for the proteolytic cleavage is the insulin. So let's take another very good example friends, that is the collagen. You all people very commonly know that. What is collagen friends? Collagen is the most important structural protein present in the body. So we all know, right? The abundant protein present in the animal kingdom, or the most important protein in the animal kingdom or in the animal body, is the collagen only. Because friends, it is going to give great structural support. We all people know that, friends. Collagen is present in the, everyone listen, collagen is present on the tendons, ligaments, joints, bones. Even friends, they are present in the smooth muscles like arteries also, even in the heart also. Most importantly, we read in the unit 4 also, they are also present in the ECM, extracellular matrix protein also. So collagen is present everywhere in the body, and majorly what it is doing, it is majorly providing the structural support. So don't confuse with that. So friends, we all know, right? Collagen is what kind of a protein, friends? Listen everyone, collagen is a triple helical protein. Everyone listen to me carefully. Collagen is a triple helical protein. And we all people commonly know that, friends. Focus every one of you on the diagram. Every protein having what kind of domains, friends? All of you. Every protein having N-terminal domain, and friends, every protein having C-terminal domain. There is no doubt about it. And every one of you has to understand, friends, at the N-terminal, listen to me carefully, friends. At the N-terminal domain of the collagen, certain extra sequences are present which do not have biological activity. Similarly, friends, at the C-terminal domain also, there are certain, what are present, friends, certain extra sequences are present even in the C terminal domain also. So friends, we have to cut this particular extra C terminal domain sequences. We have to cut this extra N terminal domain sequences also. Then only, friends, the polypeptide will become functionally active. The polypeptide will become, the polypeptide collagen will become biologically active. So friends, every one of you listen to me very carefully. How can we cut? Everyone listen to me. How can we cut N-terminal domain of the collagen? All of you pay attention, friends. We can cut this particular N-terminal domain of the collagen by a specialized enzyme. We can call, friends, aminoprocollagen peptidase. Kindly don't forget this. And here you can see, right, the C terminal domain can be cleaved by what, friends? Carboxyprocollagen peptidase. So there are total two type of enzymes are there which can cut at N terminal domain also, C terminal domain also, then make entire or remaining collagen to become biologically active. Suppose example, there is a deficiency in the aminoprocollagen peptidase enzyme. This enzyme is not present. This enzyme is not present. Then what is the point? You have to understand, we can't cleave this particular amino terminal domain. So friends, because of that, see here what happens. And look into this. An inherited, focus, an inherited defect of aminoprocollagen peptidase in the cat and sheep results in a condition, friends, we can call it dermatoxeriasis, which we can call fragile skin. Everyone listen, friends. Suppose example, in our body, the N-terminal example, in case of a sheep, let's take, there will be an absence of the aminoprocollagen peptidase enzyme. You know what happened? Then the extra sequence present on the aminoprocollagen cannot be cleaved. As a result, the entire skin will become fragile. You know, friends, if you touch the skin with a great pressure, the skin entirely comes out also. It become completely fragile. Please kindly don't forget the statement at all. And friends, the same kind of a disease, what disease? Dermatoxeriasis. The same disease can also occur in the human body. Why it is occurring in the human body? Just because of the mutations or in the defect of the aminoprocollagen peptidase enzyme. What is the name of that particular disease, friends? We will be calling it as Ehlers-Danlos syndrome 7. Ehlers-Danlos syndrome 7 is the major disease which is caused by the defect in the aminoprocollagen peptidase enzyme. Please kindly do not forget this. Okay, so I have already told you the example of the particular Prepro Insulin. You can see in the picture also a little more clearly. First A chain, B chain, C chain, what we are cleaving, you can see, friends, there are two proteolytic modifications occur. In first proteolytic modification, I have removed the signal sequence. In the second so upon removing the signal, Prepro Insulin converted into proinsulin. Then upon the signal sequence, upon the cleavage or the proteolysis of the C-terminal domain, the proinsulin ultimately converted into mature insulin. So friends, this is the first type of a process. First type of a post-translational modification. Focus everyone. That is what exactly, friends, we will be calling it as the proteolytic cleavage. Please kindly don't forget that. What is the next type of modification, friends? Next type of the modification is the covalent modification. See, friends, when I use a word covalent modification, every one of you has to remember, covalent modification means it's not a one or two kinds of a covalent modifications. See, friends, there are more than 150 different types of the covalent modifications are there. First of all, what do you mean by the word covalent modification? I told you, right? Different type of groups are getting added to the proteins, or different type of groups, listen to me carefully, different type of groups are getting added to the amino acids which are present in the protein. So they can, suppose, friends, this is the protein, right? N-terminal domain, C-terminal domain, and every protein having what, friends? Amino acid. You all people commonly know, right? Amino acid having what group? R group also. Yes or no? Of course, yes. So friends, in the covalent modification, both functional group, that is the amino terminal group and carboxy terminal group, added by this particular covalent modification. In addition, all of you has to remember, in addition, friends, even their side chains also. In addition, even their side chains are also get added by this particular covalent modifications. Now you can ask me one simple question. Sir, why we have to do the covalent modification? Why we have done the proteolysis? I have told you proteolytic cleavage, why we have done? I told you a simple point. To convert biologically inactive form of proteins into the active form of the protein. Same way, there is no change here. So covalent modifications are also occur. Why, friends? To alter the protein activity, to increase the lifespan of the protein. That is most important actually. So friends, all of you listen to me carefully. Just now I used a word that there are more than 150 different type of the covalent modifications are present. Okay, so friends, what amino acids commonly undergo this particular covalent modifications? Everyone listen to me carefully. Alanine, glycine, isoleucine, leucine, methionine and valine. These are the major type of amino acid undergo this particular covalent modification. What are the best examples of the covalent modifications? I want you people to remember this without fail, friends. Acetylation, adding of the acetyl group. Glycosylation, we already read. Addition of the sugar, we can call glycosylation, which increase their lifespan. Hydroxylation, addition of the OH group, methylation, addition of the methyl group, nucleotidylation, phosphorylation, and ADP ribosylation. Friends, this whatever the 7 or 8 process I kept here, they are more important, but what I conveyed here, there are more than 150 different types of covalent modifications are present. Please kindly don't forget it. Now all of you listen to me very carefully, friends. Acetylation. Now you can ask me one very important question. Sir, why we are adding the acetyl group to the amino acid? As I already told you, covalent modification. Friends, whenever you add the acetyl group to a N-terminal domain of the protein, friends, remember everyone, they will control the lifespan. All the students listen my statement. Whatever the protein, listen to me carefully, whatever the protein that is not added by the acetyl group, friends, that particular proteins can be rapidly degraded, very fast and very rapidly degraded. ORR, if that particular protein is not acetylated, it is not added by the acetyl group, so friends, it cannot degrade at all, right? So see here, controls the lifespan of protein within the cell, because everyone listen, non-acetylated proteins. What do you mean by the word non-acetylated? The protein was not added by the acetyl group. Since it is not added by the acetyl group, many kind of a degradative enzymes, we all commonly know that. What that degradative enzymes will do? They will come and rapidly degrade this particular proteases. So that's why in a one line I can say, addition of the acetyl group increase the lifespan of a protein. And friends, the most common type of modification undergone by the amino acids are phosphorylation. So you might read in the cell biology chapter also many times. What are the type of amino acids that are undergoing this particular phosphorylation? Serine will undergo the phosphorylation. Threonine will undergo, tyrosine will undergo, histidine will undergo. I told you many times, what happens here? When they are all getting phosphorylated, friends, that particular protein is getting activated. I hope you all people remember, we have discussed in the eukaryotic transcription about RNA polymerase 2. I hope my friends, every one of you remember this. I told you, right, C-terminal domain of this particular RNA polymerase enzyme contain 7 amino acids. If you all quickly recall, we called a heptad sequence. You people no need to worry about that. I'm just telling you, the RNA polymerase C-terminal domain contain 7 amino acid. Among those, most of the amino acid can undergo the process of phosphorylation. So friends, I told you, right, whenever this particular amino acids get phosphorylated, understand the point, then only friends RNA polymerase 2 enzyme is getting activated. And what did I also tell you? During the process of termination, I hope all of you remember, if there is a dephosphorylation occur, what happens whenever there is a dephosphorylation occur? Whenever there is a dephosphorylation occur, the protein will become inactive. So what is the statement I want to conclude? The statement I want to conclude, if the protein wants to be biologically active, the amino acids present in that particular protein has to be phosphorylated, and to make it biologically inactive, we have to do the dephosphorylation. Whatever the modifications I am conveying here with respect to a particular protein only. So what I said here, the protein, I said the protein here is the RNA polymerase 2 enzyme. Friends, these modifications will be varied from one amino acid to the another amino acid, or one protein to the another protein. Don't forget it. So friends, what we read, first kind of modification, we read the first kind of modification is acetylation. Second kind of modification we read phosphorylation. And friends, please remember, what is the third modification? third modification, friends, we can call it glycosylation. That is addition of the sugar. You have to remember, friends, when we add a sugar to the protein, so sugar also increase the biological activity of a particular protein. I hope you remember, we read in the cell biology also nice more times. There are two type of glycosylations occur, friends. What are those? One we called it as the O-linked glycosylation, friends, and another one we called, friends, N-linked glycosylation. You all commonly know that. So listen to me. O-linked glycosylation means what kind of amino acids are more commonly involved, friends? Serine is involved, as well as, friends, threonine is also involved. And friends, in case of a N-linked glycosylation, so always keep it in your mind, friends. Remembering this amino acid is very important for the exam. We get the questions from here only. That is, which amino acids are involved in the what kind of a cleavage. So what is the next one? Asparagine is the amino acid, friends, which are involved in the N-linked glycosylation. Please kindly don't forget that. And if you take a protein called as collagen, collagen contain proline, collagen contain lysine also. So friends, this particular proline and lysine, they are undergoing the modification of hydroxylation. And if you take any kind of a protein, the histidine present in that particular protein undergo the process of methylation. Prothrombin, which is an important blood clotting factor, it will undergo the process of extra carboxylation of its amino acid glutamate. So I'll quickly summarize. Amino acids present in the proteins, they undergo different kinds of modifications to make the protein biologically active. Among all those, there are more than 150 kind of covalent modifications are present. One is the acetylation, phosphorylation, glycosylation, hydroxylation, methylation, and gamma carboxylation. These are all the different types of the modifications. Please kindly don't forget it. So friends, look into this. 99% you people no need to remember the structures, but I just drawn here, that's all. Lysine. Lysine is the amino acid added by what specialized group, friends? It is added by the acetyl group. That's why we can call it acetyl lysine. And I already told you, serine and threonine are the amino acids which undergo the process of phosphorylation. So you only kindly look into this. This particular serine is added by the phosphate group. Hydroxyproline. So proline amino acid, which is present abundantly in the collagen, it is added by this particular hydroxyl group. Methyl histidine. So the histidine amino acid can be added by an extra methyl group. And if you look into this, gamma carboxyglutamate. So what you can see, friends, the gamma carbon is also added by what, friends? Extra carboxyl group. That is what exactly we can call gamma carboxyglutamate. So friends, these are all what? The best examples of the covalent modifications. So friends, that's about what? Post-translational modifications. Friends, this particular post-translational modification is a very small topic, friends, but it's very important. You people has to remember all the information, especially for the part B level questions. So friends, so far what we discussed in this particular chapter, we finished replication, we finished transcription, we finished translation in case of a prokaryotic organism also, and in case of a eukaryotic organism also. We also read post-transcriptional processes we finished, and we also finished the post-translational modifications. So friends, what are the various concepts that we are left in this particular unit? We are left with DNA repair mechanisms, this one, and friends, the most important one, that is the gene regulation. These are all the two major topics that we are left with. So gene regulation, we have to read the prokaryotic gene regulation. Prokaryotic gene regulation means what, friends? Majorly we have to read the operons. This is in case of a prokaryotes. But friends, when compared to the prokaryotes, our gene regulation is extremely complicated process, a very complicated process. So that eukaryotic gene regulation also we have to read. So friends, if you look into this 2022 February question paper, we got the question from the eukaryotic gene regulation for 4 marker. We got the question from the operons for the 4 marker. We got the question from the DNA repair mechanism in the 4 marker. Actually DNA repair mechanism they have asked in the genetics, but the concept is same. So you understand, from the 3 topic we got 12 marks. Then you have to imagine how important the topics are. Operons we got a straightforward question, but the question from the mutations. Eukaryotic gene regulation is also question from the mutation only, and in the DNA repair mechanism they asked the question about the differences between the nucleotide excision repair and another repair mechanism they have asked for the 4 marker. So friends, I am requesting every one of you read the 3 concept very briefly and at most depth of the concept. Okay, so for the next time also operons is very important. Even the repair mechanisms are also, friends, extremely important. We should not neglect that. Okay, and this particular DNA, see friends, all of you listen. When you are reading the operons and eukaryotic gene regulation, 100% of the concept, 100% to be understood. Nothing to read, nothing to remember, everything to be understood. But when it comes to the DNA repair mechanism, you have to 100% you have to remember also. At the same time, friends, 100% you have to understood also. Then only you can answer the questions from this particular DNA repair mechanisms. Okay, the first point, what kind of questions that we can expect in this particular DNA damages? So every one of you quickly recall, friends, in this particular concept we can get the questions from the types of repairs. First of all, what kind of repair that occurred to the DNA? We have to understand that. So I'm so sorry, not type of repair, friends, I'm so sorry, type of damage. I mean to say what kind of damages that occur to the DNA and what kind of repairs are there that are going to repair this particular damages. So type of damages that occur to the DNA, how we are going to repair that particular damage. That is what exactly, friends, we can call it as types of repair. So types of damage, types of repair. When we are reading the types of repair, friends, we have to understand what kind of enzymes involved in that particular repair mechanism. Finally, friends, we have to understand their mechanism of action. Mechanism of action. Okay, all these things we have to read in this particular DNA repair mechanism. So friends, maximum it will take 4 days to finish this entire chapter of the unit 3 as well as the unit 6. I mean to say together it will take 4 to 7 days. But I will try to finish less than 6 to 7 days only. Later we will start 3 chapters at a time. Okay, now focus. So first of all, we have to remember all the type of damages. I mean to say what kind of damages occurred in the DNA more commonly. Later we will come to the type of repair mechanisms. Everyone pay attention, friends. Deep urination, deamination, oxidation, alkylation, dimer formation, and friends, mutations caused by the base analog and mutations caused by the intercalating agents. These are various types of DNA damages. Now, all of you please listen to me very carefully, friends. We will discuss one by one repair elaboratively. Just pay attention. I will explain one by one. First one, depurination. Very simple point. You know, you can call it depurination also, you can call it apurination also. Anything is same. What is happening, I can tell you in one common line. There is a loss of purine bases. When there is a, so you can ask me, sir, how does this particular purine base can be lost? Just because of the cleavage of what kind of a bond, friends? N-glycosidic bond. Every one of you, please kindly look into the diagram carefully. We read the structure of the nucleotide from the 11th standard. So this is a nucleotide. What does the nucleotide consist of? Nucleotide consists of the base, consists of a sugar, consists of the phosphate group. These three together we will be calling it as one nucleotide. We all people commonly know that the first carbon of the sugar connected to the nitrogenous base via an important bond. What do you call it, that particular bond, friends? We can call it as N-glycosidic bond. So friends, what is depurination or apurination, friends? Cleaving this particular glycosidic bond. What, friends? Cleavage of the glycosidic bond. You only try to understand, my friends. Whenever there is a cleavage of the glycosidic bond which is coming out completely, the base comes out. Simple point. So that is only what do we can call it. Now once the base came out, see here, base got removed out. Now see here, friends, there is a complete gap was there. That is only we can call it depurination. At the same time, friends, we can call it as the apurination. We can call depurination or the apurination. Friends, we can also call it as the abasic site. And some students will get a doubt. Sir, does this base removal is only occurring in the purine? Does the base removal is not occurring in the pyrimidine? It's not like that. The base removal can also occur in the pyrimidine also. When a pyrimidine get removed out, then we can call it epirimidinic site. So what is the point you have to remember is, when compared to the pyrimidines, friends, listen to me. When compared to the pyrimidines, purines can be divided more in number. Always remember that point. Now another, see, once we are reading the DNA repair mechanisms, you will get a clear-cut idea how we can repair them. But right now we are understanding only the type of damages. Friends, this particular depurination, that can be majorly repaired by an important pathway. What do you call it, that pathway? Base excision repair pathway, or the BER pathway. This is the major pathway, friends, involved in the process of depurination. Next one, deamination. Very dangerous kind of mutation, friends. You know, I am not saying, friends, transamination. Transamination is completely different. Deamination is completely different. What is happening in the deamination, friends? We are removing the amino group completely. Friends, please listen to me very carefully. Listen to me carefully. Upon removal of the amino group, one base can be completely converted into unnatural bases. So please listen to me very carefully. In the human body, deamination takes place in the liver. It is the process by which amino acids are completely broken down. Every one of you look into the picture carefully. I am requesting all of you, friends, to remember the 4 examples without fail. So once we asked this question in the part B section also, just listen to me carefully. What we were doing, friends, we were removing this particular amino group. All of you look into this. This is the cytosine. Cytosine contains what group? Amine group. All of you listen to me. The removal of the amine group from the cytosine, it is converted into uracil. Now friends, next one, 5-methyl cytosine. And friends, why I am using a word 5-methyl cytosine, friends? Because friends, at the 5th position it is added by the methyl group. What friends? At the fifth position, it is added by the methyl group. That's why we are calling it as the 5-methyl cytosine. Okay, upon removal of this particular amino group, 5-methyl cytosine converted into thymine. Please kindly don't forget this. Next one friends, adenine. Adenine also upon removal of the amino group, it is converted into hypoxanthine. Please don't forget that. Next one friends, guanine. The guanine also upon removal of the amino group, friends, it is actually converted into xanthine. Now what is the point I want all students has to remember? The point every one of you has to remember, you only try to understand uracil, thymine, hypoxanthine, xanthine. Do you think all these will exist in the DNA? Not at all. DNA contain only adenine, guanine, thymine, cytosine. Only these 4 bases are present in the DNA. That's all. We can't see uracil, thymine, hypoxanthine and xanthine. What are all these? Unnatural bases. Once again listen the statement. We call it them as the unnatural bases. So friends, obviously if we don't remove this particular unnatural bases, they will move to the next generation. It become permanent, causes mutation, even the dangerous type of cancers also. That is what? Deamination. Due to the removal of the amino group, which leads to the formation of an unnatural bases in the DNA, which leads to the different type of mutations. So first one is depurination, second one is the deamination, and the third one friends, oxidation. Come on, be patient. What you have to understand by this particular oxidation friends? Very simple. Treating of the DNA with an oxidizing agent. So what happens when we treat the DNA with the oxidating agents? Of course friends, the entire base sequence will get changed completely. It's also very dangerous friends. Oxidative DNA damage is an inevitable consequence of cellular metabolism with a propensity for increased levels following toxic insult. So that's what I can use a word when compared to, listen to me, when compared to the depurination and deamination, oxidation is more dangerous type of mutation. Deamination and depurination also very dangerous type of mutations only, but oxidation is more dangerous than those two. What is oxidation here friends? Very simple. We are treating, what we are doing, we are treating our DNA bases with certain oxidizing agents. And the question also asked in the exam, that is about the 8-oxoguanine. 8-oxoguanine we can also call as 8-oxo-7,8-dihydroguanine. You people no need to remember the examples at all. I mean to say structures at all. Now look into the picture. This is the guanine. Originally friends, guanine has to paired with what? Originally guanine has to paired with the cytosine. But what happened now? The guanine exposure to the oxidative stress conditions. Because of that, what is happening? A powerful oxidizing agent. See here, at the 8th position, there is a addition of the oxo group. So that's what we can call 8-oxoguanine. Originally, guanine has to pair with the cytosine, but friends, 8-oxoguanine never ever pair with the cytosine. It may pair with the another base, which leads to the complete mutation. Next type of damages. I don't know what you do. It is your responsibility to remember every single type of the damage without type of repair without fail. So next one friends, alkylation. Anyway, name itself indicating the treating of the DNA with different type of the alkylating agents to damage that particular bases. What are the best example of either alkylating or the methylating agents? There are many type of agents are there. Methylating agents, what they will do, majorly they will add the methyl group. Anyway, Example, methyl nitrosourea, nitrosoamine, and remember only this, drugs like temozolomide. So temozolomide, methyl nitrosourea, nitrosoamines, they are the best example. Another very best example for this particular alkylating agent, that is NH2OH. Everyone listen, the powerful alkylating agent hydroxylamine. So it will change entire conformation in the bases. Every one of you please listen to me very carefully, friends. Cytosine, originally with the help of 3 hydrogen bonds, it has to be paired with the guanine. So friends, whenever the cytosine is start pairing with the guanine, immediately what is happening, friends, all of you has to understand this. Upon treating with this, all of you look into this OH group, where it is added, the OH group is added to the amino group. You don't need to remember the structures at all, friends, but I am telling you what happened. This hydroxyl group added at the amino group. Then what do we call it as? I called it as hydroxyl aminocytosine. What, friends? I can call it as a hydroxyl aminocytosine. So here cytosine originally paired with the guanine, but because of the addition of this particular OH group, it is converted into hydroxyl aminocytosine. Hydroxyl aminocytosine never ever paired with the guanine. It's always paired with the adenine. So friends, which is a very dangerous mutation because it changes the entire base transition, which means it has changed everything from CG transition to the TA transition. Please kindly don't forget the statement. Next one, friends, you can see here guanine. Guanine treated with a particular alkylating agent, we can call it MMS, methyl methane sulfonate. What do you call, friends? Methyl methane sulfonate. Upon treating with this, look into the picture carefully. This is the guanine. I have treated the guanine with methyl methane sulfonate. So at the 6th position, which position? At the 6th position, you all can see the diagram. This MMS added the methyl group. So upon adding the methyl group, friends, guanine converted into O6 methyl guanine. Now originally guanine has to pair it with what, friends? With 3 hydrogen bonds. It starts pairing with the cytosine. But friends, since the guanine is converted into O6 methyl guanine, instead of pairing with the cytosine, friends, the guanine actually come and paired with what, friends? Thymine. Please kindly don't forget this particular statement at all. So once again, the transition converted from GC to AT. These are also very dangerous type of mutations. We have to repair as early as possible before it is passing to the next generation. Please kindly don't forget it. Then, so far, friends, what are the type of damages that we read? We read depurination, we read deamination, we read oxidation, we read treating of the bases with the alkylating and methylating agents. Then, friends, the very important one, that is what do we can call it as a dimer formation. This one, friends, if we don't repair this particular dimer formations, what it will do, it will cause a particular diseases we can call dangerous type of a skin cancers, which we can call melanomas. Melanomas are majorly caused by the not repairing of dimer formation. First of all, what is dimer formation? Every one of us commonly know that thymine molecules will generally add with what, friends? Adenine molecules. We all people commonly know that. You try to understand, friends, here 2 thymine molecules are joined with each other. Now you can ask me a question, sir, how 2 thymines are joined with each other? I want every one of you look into the picture. Here there is a double bond is present, friends, at the 5th and 6th carbon, that can form a bridge-like structure. You can see, right? A bridge-like structure. Now you can ask me, sir, why this particular bridge is getting formed? This bridge will form when the 2 thymines molecule exposure to the high intensity of UV light. Very simple concept. When two thymine molecules exposure to the high intensity of the UV light, there is a bridge will be formed between the two thymines, which we call, friends, a thymine dimer. You know, this particular thymine dimer look like a chemical called as a cyclobutane. That's why, friends, you know, this dimer we can also call it as a CPD dimer, which means cyclobutane photodimer. We can call it cyclobutane photodimer. So friends, if we don't break this bridge, listen to me carefully, if we don't break this particular bridge, the same information passes to the next generation. Obviously it leads to the what we can call the melanomas, which are a type of dangerous skin cancers. Quickly recall what happens here. Two thymines paired with each other because of a formation of a bridge which is formed by this particular UV light. So friends, there are two more damages and remaining entire repair mechanisms. I will finish it in the day after tomorrow class. And in the tomorrow class we are going to discuss the most important one, friends, that is the secondary metabolites. You know, right, this February 2022 also we got 4+2 marks from secondary metabolites, which are very direct and very straightforward. Okay, that's all for now, friends. If you have any kind of a doubts, you can ask. Is that clear for everyone? I hope it's clear for everyone. I really appreciate your patience that everyone is waking up early in the morning and attending the classes. That's really so nice of you. But I want every one of you to be regular. I want you to maintain your consistency. Okay? Okay, friends, that's all for now. I'll see you in the tomorrow morning. Please kindly keep on revising the Unit 3 and Unit 6. We're gonna finish soon and you people has to write the exam. Okay. Okay, friends. Thank you all. Thank you. I will see you in the tomorrow morning class. Bye bye.